Hello and welcome to another edition of Zog Science. Today we're going to be introducing genetics and talking about kind of the basic principles that Mendel laid out through his work with pea plants. First question would be, who is Gregor Mendel? Well, he was an Austrian monk that was born in the early 1800s, and he's kind of known as the father of genetics because of the experiments and observations that he did with the inherited traits of pea plants. He worked at a monastery, and one of the things that he did was that he was attempting to figure out sort of why it was that some plants were tall and some plants were short, as well as having other traits. And so he used pretty meticulous research in order to figure that out. So what he did was he cross-pollinated pea plants, which were true breeding, and what that means is that they were producing offspring identical to the parents. So basically, if he had a short plant and he bred it with another short plant, he would always get a short plant out of it. He would never get a tall plant. Or if they had wrinkled pea pods, then they would always have wrinkled pea pods. So there were 7 different traits that he actually investigated when he was doing his pea plants. So what is a trait? Well, a trait is a specific characteristic that varies from one individual to the next. In the case of pea plants, he was looking at things like what color was the peas for the pea plants, what was the color of the flowers, how tall they were, several different things. So here we go. So yeah, seed shape, seed color, pod shape, pod color, flower position, and plant height. You don't need to memorize those. Those are just the 7 that he looked at. The thing is that whenever he was doing his studies, in order to make sure that he was not going to be messing with his data, he examined one trait at a time. So he would only look at seed shape, and then he would only look at another one. So it helped his results in order because he was doing that. He was using good scientific methods. So these are kind of the different traits and what they looked like. You kind of see the difference between them. So this is the experiment that he did. So he wanted to fertilize seeds by joining male and female gametes, right? So plant equivalent of sperm and eggs, and he would do it from 2 different true breeding plants. So he took tall and short plants. And to prevent self-pollination, because a lot of plants, what they'll do is that they can self-pollinate, he would cut away the pollen-bearing male parts from one plant, and then he would take the pollen from the other plant and move it onto the first one. This is called cross-pollination. So again, he would cut away the male portion and then transfer the pollen over with the painting brush, and you can kind of see why not many people had done this at this point, because this was a very meticulous process. So he began with tall plants and short plants and true breeding, right? So again, they were only going to produce tall shorts if they had been bred with each other. He crossbreeds those, okay, and he calls this the parent generation. So we've got a tall and a short, okay? And the offspring, the F1 generation, okay, they were all tall. Every time that he did this, he would always get all tall plants. And these are the cross between parents with different traits are known as hybrids. So the F1 generation, which is the first offspring generation, those are going to be our all talls. He then takes 2 of the F1 generation and crossbreeds those, or self-pollinates them. So basically he's got 2 F1 plants, and he crosses the F1 plants to create the F2 generation. And when he gets the F2 generation, okay, some were tall and some were short, and this was always occurring in a 3-to-1, 3 talls to 1 short. And no matter which traits he looked at, whether or not it was being tall or the seed shapes or whatever, any of those 7 traits that he looked at, he always got this exact ratio of 3-to-1. Now, there's some controversy because his data was very, very exactly on this 3-to-1 ratio, that it was possible that he might have doctored the data a little bit, but no one really knows, and it's never really been proven. But that would be pretty amazing that he did doctor the data, because at this point no one actually knew what the data was supposed to be like. So based on Mendel's work, we now know that a gene is a unit that determines a trait. Okay, so we've kind of talked about genes before as being segments of DNA. At the time when Mendel was doing his work in the 1800s, no one knew that DNA actually was the carrier of genetic information, right? So based on his work, they kind of were able to go through that. and come up with this idea of a gene. And then we have ideas called alleles, okay? An allele is a different form of a gene. So if our gene was eye color, the different alleles would be brown, blue, green, or hazel eyes. Or if we were talking about the pea plants, if we were talking about height being the gene, then the different alleles would be either tall or short, okay? So those are our different DNA sequences that could give us either being taller or shorter. And we basically have three basic principles based off of Mendel's results. Okay, in general, okay, and we're going to learn about exceptions to this rule, but at the time he didn't know about these. The principle of dominance: one allele is dominant to another. Okay, so we've got a recessive allele and a dominant allele. The recessive allele is not going to be expressed. We're not going to be able to see the results if there is a dominant allele present. Okay, and when we write these out, we're going to write them out with a lowercase letter. So remember, we've got pairs of chromosomes, right? Each pair of chromosomes is going to have one allele on it. So if we have a recessive trait, then it would be TT, little t, little t. Okay, so short, for a pea plant to be short, it has to have on both chromosomes a recessive allele, a little t and a little t. The dominant allele is always expressed even if it's combined with a recessive allele. Okay, it's going to mask it. It kind of overrides it. This is expressed with a capital letter. So tall, our tall plants could be one of two things: either big T or big T little t, right? Depending upon whether or not they are, they've got the two big T's or the big T and little t. Now, when we're looking at this, okay, the parent generation, okay, they were big T, right? Because they were true breeding. So if you took big T and you crossed it with big T, the only possible combinations that you could get are big T and big T, right? The F1 generation, those were these right here, the hybrids, big T, little t, right? Okay, the combination of alleles is called the genotype. So those letters that we were talking about, those are the genotype. That's the genes that you have, okay? If both alleles are the same, then we call that being homozygous. So big T, and little t, those are homozygous, all right? And it would either be homozygous dominant or homozygous recessive based on whether or not you had the dominant or recessive alleles. If the alleles are different, the individual is considered to be heterozygous, right? So big T, little t would be heterozygous. And then the physical appearance, or what you look like, that is the phenotype. So if my genotype is big T, then my phenotype would be tall. If my genotype was little t, then my phenotype would be short. All right, and the principle of segregation, okay, when gametes form, so gametes, we're talking about sperm and egg cells, the alleles must separate from one another, okay? So each gamete is only going to have a single allele for each gene. So let's take a look at what that kind of looks like, okay? So this is our parent generation, right? This is our parent generation. The possible gametes that we can form here are what? Big T, right, and big T. Over here we can get little t and little t. Those are the only possible gametes that we could have, right? Now, for the F1 generation, right, these can form in any combination, right? So we could get big T, or big T, little t. Those are the only possible combinations that we can have. Now, if we take these two individuals, right, the possible gametes that we can get are big T and little t, or big T and little t, right? And then we can form those in any combination, big T, little t, right? If we cross these, and then little t. And that would be our F2 generation, right? If this was tall, right, these three would be tall, and this one would be short. Hey, look at that. That's our 3-to-1 ratio of tall to short plants that Mendel was always getting. Okay, right? So we could fill that out. Move forward. Okay, so there's another way that we can look at this, and that is through beautiful things, which I'm sure you all have learned about, called Punnett squares, right? So that F1 generation, we took a big T, right, and we crossed it with little t, right? And so the possible combinations that we were able to get was right here. Okay, so you can see, right, the big T's come down, and the little t's go across, and so we are able to fill this out, right? So then for the F2 generation, we took big T, little t, right, and we cross them, these are the possible combinations that we get, right? This one's going to be tall, or these ones are all going to be dominant, and this one is going to be recessive, right? So for the phenotypic ratio, right, it would be 3 to 1, tall to short. The genotypic ratio would be 1 to 2 to 1, okay? Right, the genotypic ratio, 1 TT, 2 big T little t's, and then 1 little t little t. All right, and there's the parents in the F1 and then the F2. So the final principle is the principle of independent assortment. So when examining two different traits at the same time, for example, height and seed color, okay, you would get all the possible combinations expressed in the F2 generation. So what that means is that individual traits are inherited independently of each other, so that there is no relation between height and seed color, right? So being tall does not make you have green seeds. You can be equally either green seed or white seed. They are not related to one another. Now, as with the rest of these kind of three principles that we've gotten from Mendel, this is generally true. However, it is not 100% true, right? Because if you have a trait that is on the same chromosome, it's going to be more likely that for that trait to be linked to another trait or attached to it, right? And not be necessarily inherited independently from it. But that just kind of depends on a little bit more complicated stuff that you would learn in AP biology. All right, so I'm going to have a link for this YouTube video right here, as well as an animation. The animation goes with the independent assortment. Video is just kind of a fun one. Kind of talks a little bit about Mendel and how he never really actually got full credit for his work until after he was dead. All right, so thanks for watching, and I'll see y'all next time.